My name is Gonca. I'm a second-year cardiology fellow at the University Hospital in Zurich, and I would now like to present you our study, which is entitled "An autoantibody profile detects Brugada syndrome and identifies abnormally expressed myocardial proteins." So Brugada syndrome is known to be associated with life-threatening ventricular arrhythmias, but diagnosis can be challenging due to the transient nature of the EKG findings. As evidence is becoming clearer that myocardial inflammation may be present in Brugada syndrome, we sought to identify whether myocardial autoantibodies can be detected as a biomarker for this rare disease. For this reason, samples of myocardium were separated by 2D gels, as you can see here in this silver stain, then blotted to membranes, and were exposed to sera of 3 Brugada syndrome patients, forming the discovery cohort from Toronto. This revealed the presence of autoantibodies to 4 specific proteins. When we analyzed these 4 proteins by liquid chromatography tandem mass spectrometry, we identified them as alpha cardiac actin, as alpha skeletal actin, and keratin 24, which are all cytoskeletal proteins, and also connexin 43, which is a gap junction proteins. Autoantibodies to these 4 proteins were also consistently identified in the validation cohort of our Brugada syndrome patients in Zurich, and when absent in control sera. Notably, these antibodies were also absent in patients with hypertrophic, dilated, and arrhythmogenic right ventricular cardiomyopathy, suggesting a high specificity for Brugada syndrome. The data for these cardiomyopathies is not shown here. Next, we have also confirmed these findings by Western blots against recombinant proteins, as well as in ELISA assays, reproducibly demonstrating positive autoantibodies in Brugada syndrome patients, but none in the control samples. Next, to assess whether Brugada syndrome patient sera also binds to target proteins in cardiac tissue, we double-strained normal cardiac tissue with Brugada syndrome patient sera, as you can see in this red channel, then with commercially sourced antibodies against these target proteins in the green channel, where we could clearly see co-staining for these proteins. Finally, the myocardium from a BRS decedent and RVOT biopsies from 9 Brugada syndrome patients were assessed with each of the proteins, demonstrating an abnormal aggregate within the sarcoplasm of Brugada syndrome patients, as compared to the normal tissue control samples where they have a more filament and reticular structure. So we have hypothesized that these proteins might be misexpressed in the cells and form aggregates within cardiomyocytes, affecting the ion channel trafficking in the cell as part of the pathophysiology of Brugada syndrome. So these aggregates are then released out of the cell, where they form as epitopes for the immune system, and autoantibodies can be formed against these proteins, which are then creating a biomarker profile for this disease. In conclusion, our data so far suggests that this 4 biomarker profile is highly sensitive and specific for Brugada syndrome, but further assessment is required among other myocardial diseases to confirm its specificity. So thank you so much for watching, and now it's time for me to get inspired for further research. Thank you.